Hello and welcome back to my channel. So today's topic is nucleic acid, and particularly in this video we will be discussing about nucleotides. So nucleic acids, because they are monomers of nucleic acids, so we will discuss a little bit about nucleic acids first. So nucleic acids, they were discovered by Friedrich Mesher from the nuclei of the pus cells, leukocytes, from discarded surgical bandages, and they called it nuclein. And nuclein was later shown to be a mixture of a basic protein and a phosphorus containing organic acid, now called nucleic acid. So there are two type of nucleic acids. We also call them polynucleotides because they are made up of their monomers. The monomers are nucleotides, DNA, RNA and DNA. Ribonucleic acid and deoxyribonucleic acid. Nucleotides are monomers of both. So the monomeric units of nucleic acids are called nucleotides, and nucleic acids therefore are also called polynucleotides. Nucleotides are phosphate esters of nucleosides and made up of three components: nitrogenous base, a base that has a nitrogen atom along with carbon, a 5 carbon sugar which we usually call pentoses, an ion of phosphoric acid. So we can have one phosphorus, two or three. Next, in the nucleotides composition, we have nitrogenous bases. They are heterocyclic, planar, and relatively water soluble aromatic molecules. There are two general DNA and RNA, purines and pyrimidines. So purines are 9-membered ring because they have 9 members, carbon and nitrogen, whereas pyrimidine, they have 6-membered ring starting from nitrogen, 1 2 3 4 5 6, containing carbon and nitrogen. So they are composed of carbon and nitrogen, and they are found in DNA as well as in RNA. Adenine, also called 6-aminopurine, and guanine, also called 6-oxy-2-aminopurine. Adenine has a amino group on the C6 carbon, whereas guanine has at the C2 carbon. Adenine contains this ammonia at C6, you can see here, whereas guanine, it contains at the C2 carbon, and a carbonyl group is there at the C6 carbon. Whereas in case of pyrimidines, the two major pyrimidine bases which are found in DNA, they are thiamine, also called 5-methyl-2,4-dioxypyrimidine, and cytosine, 2-oxy-4-aminopyrimidine. The difference between thiamine and cytosine is we have an ammonia group at the C6 carbon here, we can see. In RNA, we have another nitrogenous base called uracil, which is 2,4-dioxypyrimidine. It differs from thiamine just by a methyl group. So thiamine and uracil, they differ from each other by a methyl group. Next, we have sugars. Naturally occurring nucleic acids, they have two type of pentose sugars, ribose and deoxyribose. Riboses and deoxyriboses both are 5 carbon compounds. Ribose sugar is found in RNA. Beta D-ribose is a 5 carbon sugar with a hydroxyl group on each carbon. The carbon atoms of the ribose, deoxyribose present in nucleotides, they are designated with a prime mark so that we can distinguish them from the backbone numbering in the bases, in the nitrogenous bases. Deoxyribose sugar is found in DNA, and the hydroxyl group at 2' position of ribose sugar is replaced by hydrogen. That's why we call it 2' deoxy. Next, in this, we have to discuss about nucleosides. So sugar and nitrogenous bases, they join to form nucleosides. So ribose sugar or deoxyribose sugar plus nitrogenous base, they form nucleoside. The bond between the sugar and the base is called the glycosidic bond, and this bond is said to be in the beta configuration, configuration with respect to the ribose sugar. The beta glycosidic bond in case of purines and pyrimidines both. The base is free to rotate around the glycosidic bond. Due to rotation of the glycosidic bond, two different conformations are possible. So the two standard conformations of the base around the glycosidic bond are syn and nt in case of purines. This is the syn conformation and this is nt conformation. Whereas pyrimidines, they tend to adopt the nt conformation only, exclusively because of the steric hindrance between oxygen and carbon in the syn conformation. Nucleotides, they are phosphoric acid esters of nucleosides with phosphate at C5 prime position. And nucleotide can have 1, 2, or 3 phosphate groups designated as alpha, beta, gamma for the first, second, and third phosphoric acid respectively. Nucleotides are found primarily in as the monomeric units comprising of the major nucleic acids of the cell, RNA and DNA. And there are many other functions which nucleotides perform. For example, they have a role in formation of energy currency. They can act as energy currency like ATP and GTP. They act as a precursor for several important coenzymes like NAD+, NADP, FAD, and coenzyme A. Also, they serve as precursor for secondary messengers like cyclic AMP, cyclic GMP precursors. One important nucleotide which we will be discussing is ATP. ATP is the chemical link between catabolism and anabolism, and it is the energy currency of the living cells. It also acts as a donor of high energy phosphate and consists of an adenosine moiety to which 3 phosphoryl groups are attached sequentially, linked via phosphoester bond by 2 phosphoanhydride bonds, and it's also referred to as high energy bond. So the active form of ATP is usually a complex of ATP with magnesium and manganese. So this is the phosphoanhydride and the phosphoester bond. The adenosine molecule, adenosine is ribose plus sugar, ribose sugar plus nitrogenous base, and when we attach phosphate group to it, so it becomes NTP. So in this case it is ATP. It bonds via phosphoester bond, and followed by the bonds are phosphoanhydride bonds. The table which is given below, it basically classifies nucleosides and nucleotides according to their names. So purines and pyrimidines, first of all, in case of purines we have adenine and guanine. So the nucleosides in case of RNA, in adenine it's called adenosine, and in case of guanine it's guanosine, for cytosine cytidine and uridine. In DNA it's deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. Nucleotides in RNA are called adenylate, guanylate, cytidylate and uridylate. Whereas nucleotides in DNA, they are called deoxythymidylate because they are deoxy D bases. So thymidylate, it's actually deoxythymidylate. And we have deoxyadenylate. So we also call it D-adenylate, D-guanylate and D-cytidylate. Similarly, for NMP, that is nucleoside monophosphate, AMP, GMP, CMP, UMP or TMP. For NDPs, nucleoside diphosphates, ADP, GDP, CDP, UDP and TDP. For NTP, we have ATP, GTP, CTP, UTP and TTP. So ATP will be adenosine triphosphate, guanosine triphosphate and so on. So these are the nucleotides and nucleosides of different purines and pyrimidines. Next, how these nucleotides combine to form a nucleic acid. So we call them polynucleotides. They are formed by condensation of 2 or more nucleotides, and the condensation mostly occurs between the alcohol of a 5' phosphate of one nucleotide and 3' hydroxyl of the Second, with the elimination of H2O from a phosphodiester bond, and all nucleotides in polynucleotide chain, they have same relative orientation. This is an example of a polynucleotide. Guanine, adenine, thymine, and cytosine groups are there in 5' to 3' direction. So the primary structure of DNA and RNA, the linear arrangement of the nucleotide looks like this only, which proceeds in 5' to 3' direction. And the common representation is given here of the primary structure of DNA and RNA. This is a sugar attached to a phosphate molecule, and the bond is called the phosphodiester bond. Again, a sugar attached to a, this is a CH2OH sugar and attached to a phosphate group, again a phosphodiester bond. So these are the phosphodiester bonds which are mentioned here, by which a polynucleotide structure is stabilized. The order in which the bases occur in a polynucleotide describes the primary structure, and the convention is to list them left to right from 5' to 3' end. So this is how primary and secondary structures of DNA and RNA molecule they are constructed. For DNA, the most important secondary structure consists of a double helix, two strands held together by hydrogen bonding between the bases, and we will discuss that in our next video. Till then, thanks for watching. Do subscribe to my channel, and thank you.